Good evening, everyone. This is Dr. Prasanna, and I profusely thank Barcode Biotechnology and HydraGen Private Limited for this invited lecture on research today on current trends in Hydra research, the tools, trends, and transformation. So this is jointly sponsored by both HydraGen and Barcode Biotechnology. What are you seeing in the right side of the window is actual live Hydra captured in HydraGen Private Limited lab. They are feeding upon Artemia. You can see Artemia sticking on to the tentacles of Hydra and immediately paralyzed, and through the mouth region, it is being engulfed inside. You can see the Artemia being captured and eaten alive by Hydra. Okay. So for those who are very new to this topic, what is Hydra? Hydras are very simple organisms. They are nidarians, freshwater invertebrates, few millimeter in size. They are simple yet very powerful organism. Okay. So they have a simple body plan. If you see the body of Hydra, but it has become one of the most influential model in biology right now because of its regenerative ability. They are animals, but they can regenerate. Every fragment of Hydra can become an entire organism. So they are used in developmental biology, toxicology, and they are not known to die because of aging. They are literally immortal organism. Okay, so because of this extraordinary regenerative capacity, they are actually the entire body since can be generated from a tiny fragment. This unique ability makes them essential in understanding their, tissue plasticity or pattern formation, the mechanism that prevents aging in Hydra. Okay, so this Hydra, what are you seeing right now is from HydraGen Private Limited here. We have a Hydra vulgaris. We are using Hydra vulgaris for various toxicological and other assays, especially in ecotoxicological assay. They are very much used, and even in neurological assay because they have a simple nerve net structure. They don't have a well-developed neurons. They have a simple nerve net structures. So they are used in various neurotoxicological assay too. So if you see the Hydra's body plan, you can simply segregate Hydra into head region, body region, and foot region or peduncle. Okay, so they do have a tentacles. Tentacles are actually accumulated with a nematophore or nematocyst. This is the apparatus through mechanical force along with the toxin. They can paralyze the prey and they can take it in. What you have seen here is actually a hydra being fed live with Artemia, a microcrustacean, Artemia naples. So when they stick on to the tentacle, you can see a nematocyst in the tentacle region here. You can see when the Artemia stick on to the tentacle, they are instantly paralyzed, and paralyzed Artemia will be engulfed through the mouth region of the Hydra. The entire animal is not even 2-3 millimeter in length. Okay, so because of this extraordinary ability, they've been used in various toxicological research, regenerative biology. So before getting into what is the current trend in Hydra research, I want to touch upon who is my sponsors. So this is being sponsored by Barcode Biotechnology. They are founded in 2013. They are basically a DNA sequencing company. I would say they are India's most affordable DNA sequencing services, giving both Sangus and Illumina sequencing. So basically, they use DNA sequences and the associated instrumentation to understand biological data. So they have a Megabase and ABI 3700 model in their company, and they have a couple of PCR workstations for gene-targeted sequencing. And this is a bread provider, Mermaid 192X synthesizer. This is a PCR primer synthesizing machine that actually runs 24/7. They fuels Barcode Biotechnology. Apart from this, they are known for authentication services, plant species identification, Even herbal powder authentication. They can decode the composition of plant species used for preparing a herbal powder if you send them a sample. Even in microbiology, bacterial species identification through 6ns RNA sequencing. You can even send them a mixed culture. You can find them out what are the colonies you want to get sequenced. The colonies will be sequenced, identified to the species level. You can also get a glycerol stock for further preservation of your sequenced colonies. Mushroom identification through internal transcribed region sequencing. Even animal sequences, invertebrates, vertebrates to cytochrome C oxidase 1 sequencing, multi-locus sequencing. In microbiology or in any authentication of microorganism, you may have to sequence two genes. Yeah, even in the plants, both RBCL and MATK genes are sequenced for precise species identification. So such a services are given. Even Forge identification, viral gene sequencing, insect barcoding using COIG. So these are the various services provided by Barcode Biotechnology. And if you want to know about various programs they are conducting, given it as an internship or after sequencing, many customers come up with various questions of how to analyze sequences and other stuff. You can follow this channel to see videos explaining how to analyze DNA sequences. These are various social media profiles where you can follow Barcode Biotechnology. HydraGen is a new company. They are exclusively focused on using Hydra for various biological assays. You can visit HydraGen at www.hydragen.com. This is a flagship product of HydraGen, Hydra Vault, where if you want to establish a Hydra culture in your laboratory, this is just costing us around $1,000. You can get a Hydra Vault product which consists of Artemia, Hydra, aerator, ball, 100x Hydra media, the pasture pipette, everything together. It's an instant lab. You can simply buy the product, establish a culture. If you are someone who cannot establish a culture, maintain the culture, and use it for assays, these are various service-based, business hydrogen does along with Barco Biotechnology. You can send the samples to them. You can point out what kind of assay is required, and hydrogen is very happy to provide these assays. The turnover time is usually 7 days. So let's see what are the ways Hydra can be used in the research. So here is my first proof. Here is from a recently published article where author used these keywords: Hydra, Hydra plus toxicity, toxicology, toxicological, and see the number of citations for last 180 years. See the number of citations in the published review article. It's clear research and review articles. It's clearly going up. Okay, so especially Hydra, and since you do not need ethical clearance like it is required in animal cell culture or using animals or rabbits, mice for performing various toxicological assay, Hydra is a preferred model and it's a simple model. And many of the disease-causing genes in human beings can be equated with the Hydra genome too. We will see how in further slides. Okay. So let's see the trends in Hydra. Before that, let's understand how this model organism is used in biological and ecological research. Okay, Hydra can reproduce by budding, like plants. Okay, they can simply bud out of the middle part, and the buds will fall off, and they will become a new progeny through asexual reproduction. If stressed, they can also produce gametes, and sexual reproduction is also possible. But common mode of reproduction, especially in Hydra vulgaris, that is being maintained between 18 degree and 22 degree in the lab, you will see they rapidly reproduce by asexual means. Usually, we see doubling time as 3 days. Every 3 day, Hydra will double, and the parent Hydra never dies. Okay. So the simple body plan, they just have a two epithelial layer of ectoderm and endoderm, and they are dynamic mesoglia, and they have tentacles. And in the tentacles, you can see nematocysts. There are usually four kinds of nematocysts, and the entire body has a nerve net-like mechanism that can, behaves like a loop, swings, or even somersault is done in Hydra. And the tissue stiffness pattern, if you see, they have extracellular matrix dynamics and specialized cell types. There are around 20 to 21 different types of cells. The reason for Hydra not being, very active in their life, not aging, is because of their continuous stem cell regeneration capability. Okay, they will continuously produce stem cells that can, keep on perpetuating, and they do not undergo a cycle of aging. Okay, especially these multipotent stem cells. They are distributed throughout the body, giving the extraordinary ability to regenerate the entire body. There is something called trisection assay that is done with HydraGen Private Limited, where they cut the body parts into three pieces. You will see you can treated and untreated with heavy metals or nanoparticles or any toxin compound. You can see the regenerative assay, and you can even convert the score, the length of the regeneration in the Wilby scale, and it can be equated to, assess the impact of the toxic component or pollutant on the environment. I will explain you how this is done. Okay, so basically they are actually a freshwater habitats. They are found in the pristine lakes. The hydra what we have is actually from Nepal pond. The hydra vulgar is in HydraGen Private Limited, is actually isolated from the pristine lakes from Nepal mountains. Okay, with like I said, they can adapt to varying variable conditions. If you see in evolutionary point of view, they are actually nidarians like jellyfish and sea anemone. They are actually nidarian, well characterized morphology, and you know. These are the important characters that made them, fundamental multicellular biology. If you want to understand that, Hydra become a very popular model, okay, especially the nematocyst. There are various studies talking about the protein nature of the fibrous component contained inside the nematocyst of the Hydra, which actually paralyzes the prey. Okay, so many studies, recent studies are actually happening. These are silk-like protein toxins, actually rich in cysteine and other sulfur-rich amino acids. These recent studies clearly show that their role in paralysis and taking the prey. This study went ahead and they actually isolated the gene and cloned it into the E. coli, and they expressed the nematocyst silk-like protein in E. coli. Okay, so it's a very interesting study when it comes to the nematocyst. If you are very curious about this study, I would recommend you to read this article. Okay. So in morphological studies, if you see this modern aging and the genetic tools actually greatly expanded the Hydra research. Okay, the advances such as stable transgenesis, gene silencing, genome editing, and high throughput omics actually have improved the imaging system and transformed the Hydra into a very powerful modern model, I would say. The techniques like cryofixing and high pressure freezing preserve actually the ultrastructure with exceptional details. You can see here. Here is a hydra called Hydra viridissima. They actually have a symbiont, green algae, that lives inside the tentacle of the hydra. The algae helps hydra in regeneration and maintaining the osmotic stress and many more factors. And hydra in fact act as a host, hosting this algae as a symbiont in them. And there actually this Hydra viridissima is an exact or excellent model for studying Hydra endosymbiosis, predator-prey interaction, stress tolerance, even in metal detoxification. It is very clearly proven that this viridissima, whenever they have algae, chlorella in their tentacles, their tolerance to zinc and copper toxicity is much more better when they are absent. Okay, when this algae is absent in the hydra. So they are actually becoming a ideal model for studying the stress tolerance and metal detoxification. This symbiotic algae actually help hydra survive even high temperature stress, and like I said, mitigate copper and zinc toxicity. And intern hydra actually here regulate the algae population, expels excess algae during feeding to prevent oxidative damage, and making it a key model for understanding the symbiont specificity or even population control, how this symbiosis works. Okay, so this is not simply this is not possible 20 years back, even though this animal has been studied for more than 100 years now. This is mainly possible now because of the new low cost and high resolution imaging platforms like GLUP scope, SimFlow. There are many advances that actually triggered this visualization of Hydra's 3D movements, surface attachment, regeneration mechanism, hydrodynamic behavior. Because of this cutting edge technology, these are possible right now, which is not possible 5 years back. Okay. And if you see in toxicological research, this is something I explained previously. This is called Welby scale. What is happening here is, like I said, they have a unusual regenerative capability. Every fragment of Hydra can become an entire animal. So what they will do, they will give a score of 10 for a full-fledged Hydra, and they will give a score of 0 for a disintegrated Hydra. So now treated and untreated with it, heavy metal nanoparticle or even environmental toxins, you can compare the treated Hydra with untreated Hydra and give them a score. This score can be an equivalent to assess the impact of toxic component. Higher the score, lesser the impact. Lower the score, the impact is very high negative impact can be seen for that particular compound. Okay, so if you see, it's very highly sensitive to wide range of pollutant. Okay, that's why they are always present in the pristine lake. The pollutants can include heavy metals, organic contaminants, pharmaceuticals, endocrine disruptors, nanoparticles, microplastics. For all those assays, you can very well use this model as a efficient model. Okay, so all right, and hence they can also be used as a bioindicator of an environment, and you can assess the pollution status of a particular environment. If Hydra is there, definitely it's a pristine environment. They are not yet polluted much. Okay, so they have the ability to accumulate contaminants, and also they possess human-like proto-oncogenes, allowing it to relieve genotoxicity effects. Very much, I will talk about that in future slides. There are various, other tools like alkaline comet assay, which is a popular assay, HydraGen Private Limited gives us a service where you can use a Hydra to, along with the alkaline comet assay, you can assess the genotoxicity of your pollutant or any compound that needs testing. Hence, this relevance to modern ecotoxicology is very high. And it's a central in, Hydra vulgaris especially is a central organism in assessing molecular toxicity mechanisms, stem cell impacts, or teratogenicity or endocrine disruption, and even pollutant-driven carcinogenesis. Okay. Amazingly, these organisms can also form tumors. Hence, they are actually very recently been extensively explored for tumorigenesis too. Okay. So how are they used in environmental decision making? Okay, Hydra, if you see, like I said, basically they will study the budding behavior, or trisection assay. They used to study the regenerative rate between the treated and untreated one. Even if you see over the past 2 decades, it has transformed actually the environmental assessment because of their simplicity, sensitivity and short life cycle. Like I said, in 2 days time they can double, and rapid clonal growth, and its wide distribution in diverse aquatic habitats, and along with the history of highly reliable model for testing the pollutant, assessing the organism, make Hydra as a ideal organism for environmental decision making. Okay. Uh, Including ecotoxicology or in the environmental monitoring. Okay. Another exceptional quality of Hydra is its holobiont. As a microbiologist, I should mention this. Curvy bacteria gracilis is a one species of bacteria exclusively present in Hydra vulgaris. You can't see this bacteria in any environmental genome assessment or in any other ecosystem. They are exclusively present in Hydra vulgaris. Because of its symbiotic lifestyle and well-characterized physiology, now it's an excellent model for studying holobionts also. Holobionts are organism. The entire organism of an individual host is called as an holobiont. And by studying that, you can see how these microbes collectively influences the Hydra phenotype, evolution, health, and many more. Okay. So the hologenome theory of evolution states that holobionts, host plus microbial genomes, together act as a single evolutionary unit. The long-term coexistence lead to a deep interdependence where organisms mutually regulate each other's physiology. And gene expression, adaptation, evolution, many aspects are actually co-regulated in the holobiont. Okay, so Hydra is becoming an ideal model for holobiont research also. All right. So finally, the current trend in Hydra right now is its ability, or Hydra can now be used for a model for cancer research. The main door actually opened because of this gene, WNT beta catenin signaling pathway. They are actually highly conserved across evolution, regulate major developmental process in Hydra, such as head formation, body axis, patterning related to tentacle budding and the cell proliferation. The same pathway, when this WNT is also found in humans, but when disturbed, they contribute to diseases, various diseases, including cancer, by dysregulating MYC gene in the humans. Okay. So this evolutionary route of cancer biology, I feel Hydra possesses mainly because of this MYC gene, where gene 1, 2, and 3 is there. They perform various function and co-regulate itself, and they act as a causative agent for tumor genesis. So Hydra can even develop a natural occurring tumors, highlights its value in understanding the evolution of cancer and cell cycle control. So this is something very new, and it's happening in the current research market. So these are the way. I hope you have learned something very useful because of this Hydra. We initiated with this video clip of Hydra actually engulfing Artemia directly into its mouth. That's how we feed in HydraGen Private Limited. One Hydra, 10 Artemia will be the ratio. So we saw what is Hydra, the simple body plan. They are just 2 millimeter. And, if you buy a Hydra vault, you will be given around 25 Hydra each. You can very well in 3 to 6 days time increase to 100 Hydra. We are actually beaming with, I would say HydraGen Private Limited incubates around 25,000 Hydra in more than 50 tank liter capacity. So I would say it's an India's biggest Hydra culture facility you can see. Along with the Barcode Biotechnology, which is basically a DNA sequencing company, they gives various assays, including Hydra aquatic toxicity assay. If you can get a Hydra vault and start your own lab. We also give counseling, and we give a consultancy of how to maintain Hydra lab. There are contact details in this brochure. You can contact for obtaining various services. We will see why this organism is important. You can see the number of citation that is been skyrocketing in recent times. We understood what is Hydra, their simple body plan, and what make Hydra lethal, how they are killing the prey, and interesting publications actually working on this cross-linker free nanofiber material from Hydra. And we saw about why this organism can be used for various applications, including studying symbiosis, how they are used in environmental decision making, how various components like microplastic, nanoparticles, pollutant, heavy metals can be tested in Hydra, and this regeneracy assay, how they are useful. This is how actually HydraGen performs. Whenever you can send samples to us, microplastic or any heavy metal, if you want to test, we will make it into various concentrations. We will use both treated and untreated Hydra. We will see the growth rate, regeneration capability. We will convert them into Wilby scale. we will give you a score of how to assess your compound, the effect of its effect on Hydra's growth. So we see about holobiont, how this Hydra research actually they maintain their unique microbial consortia. Even if you sample Hydra water and Hydra, you will see their microorganism is very unique in nature. The recent trend in Hydra research is especially with the tumorigenesis. Hydra is developing evolving as an ideal model for studying cancer. Whenever you want to do research in cancer, especially transcriptomics and genomics plays a vital role to understand the upregulation, downregulation of genes. So, along with this services, Barcode Biotechnology is also offering DNA sequence analysis services, where if at all you are using Hydra mRNA extraction, cDNA synthesis, and going for target-specific amplification sequencing. Once sequencing is done, whether it's a Sanger sequencing or in Illumina sequencing, understanding the sequences forms a major role. So this winter internship that is going to be initiated by Barcode Biotechnology around 17th December is an ideal opportunity. This is a 2 weeks intense program where both Sanger sequencing and Illumina sequences are actually analyzed, and participants will be trained in analyzing these sequences by themselves. After gaining these skills, you can also do a meta-analysis, download sequences from GenBank. You can have your own interpretation. These days, there are, the wet or dry lab data is getting accumulated like anything. We need more hands and incorporated with the AI to analyze the sequence data. Even this training incorporate a rug augmented AI model along with the molecular evolutionary genetic analysis softwares to train the participant in DNA sequence analysis. I will be posting the link in the WhatsApp group also. So I wish you may have more queries. The contact details are given in the brochures. If you have a query regarding this webinar, feel free to write your question in the comment section. Everyone who participate in this webinar will be given a e-certificate. It will be circulated in the group by 9 p.m. today. You will be given a certificate number in an Excel file along with your download link where you can go to the link and put your certificate respective number. You can download your e-webinar certificate. So thank you very much for joining us today. Let's use Hydra to advance our research. Thank you.